blocking compounds, the natural handbrake on leptin signaling, this SOX3, is upregulated in the vagus nerve bodies of these mice that have got the obesity. So what the problem with becoming leptin resistant does is the whole system is geared so that leptin, if it doesn't see enough of it, it tries to get you eating more or moving less. And that's true even at the level of the vagus nerve, the nerve that's talking to the guts. The satiety receptors do not work as well. See, the cholecystokinin, one of the main satiety signals that helps end your meal, doesn't work as well once the vagal nerve is resistant to leptin. More satiety signals not working as well here in the diet-induced obese mice. And here are some signals that tend to promote hunger, and these are now working much better in the leptin-resistant vagal nerves. So the interesting bit about the gut bacteria, you don't see these changes in germ-free mice. You don't see the change. You don't see the increase in food intake at the point where the nerves become leptin resistant. You don't see the inflammation in the upper gut in the germ-free mice. It's not there, and you don't see it in the colon either. So it is going on in the upper gut, and the eventual increased weight of these rodents and the altered plasma glucose and insulin levels in these rodents correlate very nicely with the level of inflammation that's seen in the upper gut, the inflammation that was dependent on the bacteria being there. So there's a few bits of evidence in humans that there's something going on that involves these gut bacteria, and that it's something in the lumen of the gut that's important. They transferred intestinal bugs from lean donors, and were able to increase the insulin sensitivity of people with metabolic syndrome, even though they looked at it 6 weeks later. So I'm surprised, because they were presumably still eating the same food in the interim period, that there was any effect still there, but it was still there to be significant. There's an interesting binding resin that I heard about in a talk called sevelamer, which is used in renal failure. I think it binds up some anions or others. It's interesting that even compared to other agents that are used for the same purpose, this sevelamer compound is able to reduce some of the biomarkers that are problematic in metabolic syndrome and others, including leptin and triglycerides and the oxidation, glycation of the hemoglobin, even though this resin doesn't get out of the gut. So it's as though something that was being produced in the gut lumen that was worsening the levels of leptin and whatever has been stopped from getting through by this resin that doesn't get out of the gut. So that's an interesting potential clue. Here's some very recent mouse data that's, to me, strongly suggestive that something's going on in the lumen, the tube of the gut itself. This is your standard diet-induced obesity model, where high-fat chows make the mice put on the most weight. There's intermediate weight gain with a high fat, high sugar, and the raw foods are resistant to bacterial decomposition, inasmuch as the cells constituting such foods are or recently were alive. There is no need to discuss here the biological mechanisms involved in this resistance. What matters is that such resistance is not present in inert foods like refined sugar. Thus, the sugar present in portions of sticky sweets lodged between the teeth will be fermented much more quickly than that present in pieces of raw fruit lodged in the same position. And at that point, because I moved from low carbing and found that it was when I got rid of all flour that the change happened, and was now eating a high carb paleo diet, the penny kind of dropped. So we have this almost spooky power of the microbes over host metabolism that we're only just beginning to understand, but it seems to be really reproducible in animal models and the early human stuff. We've got all the evidence that Western carbohydrate, the flour and sugar, seems to have a negative impact on metabolism. And when you go low carb and cut out most of this stuff, you get pronounced benefits. And we've got the evidence that the old whole foods produce great metabolic health regardless of the macronutrient makeup, just as long as the stuff's made of cells and is made of life. And Cleve's brilliant insight that bacteria may respond differently to inert simple energy sources versus complex life forms with evolved defenses. And so eventually, of course, I had to write the paper. So I'm arguing that food is more than just chemicals. It's the interplay with life and life within life, and all of it, each of them affecting the other. Ancestral foods were cellular, they were alive. Because cells tend to be 80% water, they have a lower carbohydrate density. The nutrient release is delayed from the small intestine onwards, not from the mouth onwards. They have increased complexity, the structural and molecular complexity, which plays into the evolutionary thrust and counter-thrust of animal and plant cells versus bacteria. This is an evolutionary battle these guys have been playing since before animals evolved a gut. So you can imagine that a bacterium might behave differently around a cell that's got defenses, pattern recognition molecules and stuff than it would against something that's going to passively let it eat it. Modern foods are acellular, dead chemicals. They have a higher carbohydrate density. These carbohydrates are released from the mouth onwards, so they affect the mouth bacteria too. They've got reduced complexity, and they behave much more like a simple nutrient broth just sitting there to be eaten. So instead of a millions of years old game of chess between old adversaries, we've got Homer Simpson in the land of chocolate. And that this may produce different genes being expressed, different pathways used, and over time you may wind up with different bacterial populations. So this is a demonstration from the paper that if you plot out foods, mark them as either ancestral foods that we could conceivably have been eating for a very long time, or modern foods that are grains or industrial products or processed foods, and plotted them out as to the weight of carbohydrate per 100 grams, i.e. what concentration will this form when it hits the intestine and forms a solution? What will the concentration of carbohydrate be in the gut? And you can see this sort of demarcates the two types of food quite, in a way that caloric density never really did, because meats and nuts have got plenty of calories per mass. And glycemic index, again, there's lots of overlap between the foods that you can apparently eat with relative impunity and the ones that are going to get you. So this is an overview of the hypothesis as a whole. So living cellular foods, when you eat them, you're going to have a healthy oral microbiome, and you're going to have decent dental health. It does for morning breath quite nicely, actually. And you're going to get evolutionary consistent signaling in the gut that the bacteria are used to it, you're used to it, everyone's happy, you get accurate energy homeostasis, and so you can actually trust your hunger and just get on with life. The minute you start pouring in this stuff that may be a dense nutrient broth, you get a perturbation of the mouth bacteria and possibly periodontal disease as a result of this, and tooth decay. You're going to get inflammation within the gut. You're going to start messing with the leptin sensitivity of the vagus, so you're going to be starting to eat more, and then over time that'll feed back with increasing problems with inflammation and move you towards diabetes, either as an